Okay, so this is supposed to be a tutorial, and what I was thinking that I would try to do is give you some impression of some of the very cool experimental methods that people have come up with. And I guess I want to begin by just saying, once again, a few words of background. And I want to use this, this very interesting article from 1902 as a way to introduce what I have to say. So as I say at the top, new instruments and new science. So let's see. There. So so this is basically, I'll try to be somewhat brief about it, but this is a story about the physical laboratories in Heidelberg and about the rise of astronomy. And so the essence of what goes on in this thing, it's talking about Kirchhoff and Bunsen. And you can see that it says that these guys, they were looking out a window. From this window, one may look out over the Rhine plain towards busy Mannheim, as Bunsen and Kirchhoff did one night when a fire was raging there, and they were able by spectroscopic examination of the flames to ascertain that barium and strontium were present in the burning mass. And so where this led is that there's very charming things that are said, but what's really important is that these guys got the idea that if they could look at a fire that was 20 kilometers away and figure out what was in the burning mass, why not attempt to use this to figure out what's in the burning of a star? And so they put their spectroscope on a telescope, and as the saying goes, the rest is history. And the thing is that that kind of technology development was critical to being able to measure the expansion of the universe. For example, Edwin Hubble, which is these pictures on the right. So why am I telling you this? Because as I said this morning, we're really living in a moment when the ability to query biological systems is exploding, and it's permitting us to ask questions that were unimaginable. In a way, they weren't even articulatable. I know that's probably not a word, but 100 years ago, people thought that that the genetic information was in proteins, not in DNA. So you couldn't even address a question like, what's the sequence of a nucleic acid? It wasn't even a question. It didn't even exist as a concept. So here, what I wanted to show you, this is kind of old school, but I think it's important to really bear in mind. The modern biology, molecular biology revolution depended so strongly on radioactivity. And for 30 years or so, it was absolutely critical. But nowadays, I would say probably the three things that I want to talk about are the palette of fluorescent proteins, the ability to do PCR, and the ability to do DNA sequencing. So what I'm going to do is I'm going to talk about these different kinds of things in the next 45 minutes or so. So let's start with exploiting the fluorescence palette. So this morning we already talked a little bit about using fluorescence to count things. But of course, as we saw from Ted Cox's talk, another use of these proteins is to learn things about the state of localization, which is very interesting. It's useful and interesting to know where objects are. Of course, there's dynamics. There's input-output functions. There's various methodologies that have arisen, such as FRET, which is basically a molecular ruler. It allows you to figure out with sort of sub-nanometer resolution what the proximity of different fluorophores are. And you can use that as a surrogate to figure out what's going on with the structures that you're interested in. This is fluorescence recovery, oh my gosh, FRAP. Fluorescence recovery after photobleaching, I think, is what it stands for. And this is a trick for basically turning a lousy thing, which is photobleaching. It's really an annoying fact that you photobleach molecules. But clever people like Watt Webb at Cornell realized that they could use FRAP as a trick, as a way to actually learn about dynamics. So here it is in a nutshell. I'm not going to spend any time on it, but let's say you have a field of view with a cell, and you burn a little hole of photobleached molecules in a little square. And then you look over time as the fluorescence resumes in that little square. How does that happen? Because fluorescent molecules diffuse into that region, and so you can learn something about dynamics. So for example, people have learned a lot about trafficking. And then nowadays, another thing that I just love is beating the diffraction limit. I'm not going to comment on that, but that's another area that I think has been very powerful. So the first part of what I want to talk about has to do with a specific set of questions which really focus on gene regulation. Okay, but this is just one example of the fluorescence palette. You could pick your own favorite example. The main point to take away from what I'm saying is the ability to actually use fluorescence in a way that is actually quite quantitative. So here I show you a famous diagram. It's probably something that, that Art Lander will show in the coming few days, I suspect. This is a sort of garden variety network that's associated with the specification of the endomesoderm and sea urchins. And it's designed in a way to look like an electronic wiring diagram. And so the question I'm going to pose is it a useful analogy or is it actually damaging analogy? Does it cause us to think in a certain way that's actually wrong? Okay, and we'll try to address that. But what I would like to say is that in order for this analogy to hold water, you've got to be able to go back to the various circuit elements and understand the input-output relationships in a concrete way. So if you look up that book on the left, which is kind of a classic book that all grad students in physics in the US have to encounter at one point in their lives, it has you open it up and it has all these different circuit diagrams and it has ways of computing the relationship between current and voltage and so on. And so what I want to do is I want to explore that analogy in the context of regulatory networks. Okay, so I'll talk more about this tomorrow. Today I'm just going to simply tell you the outcome of a calculation, which is the fold change. It's exactly what Ted told us about earlier. And this is for the case where you have a circuit which is basically very simple. There's a binding site for polymerase and there's a binding site for a repressor. And the repressor works by exclusion in the sense that when it's present, polymerase cannot be. Okay? Of course, this can be generalized. There's more complicated examples, but that will suffice. So you can work through this, and I will explicitly on the board tomorrow, and find that the fold change depends on the number of repressors and the binding energy of repressor to its operator. So what these curves show you is the fold change. That is, how much reduction in gene expression do you get as a function of the number of repressors. So let's just think about that intuitively. If I increase the number of repressors in a cell, I would expect to get less expression of my gene. So the fold change should go down. Notice that this is 1, and then this is 10 to the minus 1. So I get 1/10, 1/100th, 1/1000th. So as I increase the number of repressors, I get less expression. The hierarchy of curves corresponds to different binding energies. So if I bind repressor more strongly, I don't need as many repressors around to get the same level of repression. Okay, so that's why there's a family of curves. So what I want to do is I basically want to sort of talk about what are the knobs, if I pursue this electronic analogy. In electronic circuits, we have all sorts of knobs that we can tune. We can tune the resistance, we can tune the capacitance, we can tune the inductance. That's the kind of stuff. You can imagine in the old days, we did this with switches, or they're called potentiometers. They probably don't exist anymore. But you actually turned something, and there were capacitors that passed through each other and stuff. That's the old days. So we're interested in pursuing that analogy. In other words, if I have a cell, what are all the knobs that I can own, both as a theorist and as an experimentalist? So I've told you about 2 of them, and those are the 2 that I'm going to focus on here. The fold change depends on the number of repressors and the binding energy. So what I want to say is, how would we go about testing such a thing? And this is by no means unique to us. So Ted has already told you examples. You saw today that family of curves that he showed us. It's exactly the same mentality as what I'm talking about now. So I just want to give you a few ideas about how one pursues that. So using fluorescence, the way that we measure the fold change, we or anybody, is to basically measure the level of fluorescence in the strain that has the transcription factor doing its business, and then in the strain that doesn't. You could do this either by knocking out the transcription factor or by using inducers. You could do this in microscopy or using what's called a flow cytometer. There's all sorts of different ways. But at the end of the day, what you're interested in is the mean expression in these 2 different scenarios, and then you construct the ratio. And that's what we mean by the fold change. Now, an alternative scheme, which is more old school, is to use an enzymatic assay. And actually, Tom Kuhlman, who worked with Ted, is quite an expert at these as well. So an enzymatic assay would mean that you actually produce a protein, for example, LAC-Z, beta-galactosidase, which we heard about this morning. It clips a sugar, and when it does, it actually leads to some consequence. Now, in the 50s, chemists were incredibly clever. I still just can't imagine how people are so clever. They made all sorts of different analogs of LAC-Z that do fun things when you cut them. So some of them become yellow. So I guess maybe you talked about clear white or blue clear selection and stuff. So there's all these different tricks where you cut a molecule and you get an output. So if you use ONPG with LAC-Z, you'll get increasing yellowness over time, because what you've done is you've broken open the cells. The cells have these molecules, okay? You made the cells produce the enzyme. And depending on how much gene expression there was, if there was a little gene expression, you won't have very many of these LAC-Z molecules. If there was a lot of gene expression, you will have a lot of them. And so what you do is you start a stopwatch, and you ask the question, how yellow does it get as a function of time? And the slope of this curve tells you something about how many LAC-Z molecules there were. So that's another example. And I'm just saying, these are two different ways of going after the same result. So now what I want to do is I just want to briefly walk you through two different ways of measuring this. And again, just to try and make this point about precision measurement. So the equation that we wanted to test has this R, which is number of repressors. That's what I need to know to test this equation. In other words, I showed you a graph, and on the x-axis was number of repressors. So if I have any illusions of comparing the theoretical description to experiment, I better figure out a way to measure this quantity and that quantity so I can put data points on here. So how do we measure the number of repressors? So I'm going to show you two different ways. One of them is to basically decorate a surface with antibodies to the protein we're trying to count. And then what we do is we break open the cells and paint them, or let them flow on top of the antibodies. And then we have a scheme for making them light up. And at the same time, we also use purified protein of known quantity to calibrate. In other words, you say, well, I'm going to take my calibrated amounts, and that's these guys. And then you find your arbitrary spot, and you just ask, where does it appear? And you interpolate between the nearby points. So that's the way of doing the counting. And you can do this with fairly high resolution. And question? Yeah.
How do you control number of cells?
You don't necessarily control number of cells. You just make sure you're careful about counting them. So the point is that you grow up your cells, you break them open, but before you do that, you count them. And there's many ways to count them, and we do all of them. So the stupidest thing you can do is basically pass light through the solution and look at how opaque it is. But you can also take the cells and put them under the microscope. You can use a hemocytometer. You can plate them. There's all these different things you can do, and they're not always the same.
I was confused that if it's single cell for each block.
It's not. It's not at all single cell. This is totally bulk. What I'm going to tell you about in a little while is single cell, and it'll be very interesting to compare and contrast those. And as far as the fold change goes, this was done using the enzymatic assay. And so the net result of such a thing is this. So here's the fold change as measured basically by LAC-Z. Here's the number of repressors as measured using these quantitative Western blots. And this is the level of comparison between theory and experiment that we got, Hernan Garcia got for that particular measurement. Now, I'm just going to remind you, I'm not going to belabor this, but this morning I made this big deal about at least my attraction to the idea of do different methods yield the same results? Yes? Yeah, very good. So we've done it many different ways. My favorite way was to actually go back to some very old experiments by Mueller-Hill and fit his fold change. He only had 2 data points. We get the binding energies from him, and then we just use them. Okay, that's one way of doing it. But you can do it all sorts of other ways. For example, I mean, we've done it every which way you can imagine. You could, of course, fit all the data points. And so, for example, for the blue curve, this is the kind of number that you would get. Or you could fit, you could choose one of them and fit that and then use it to predict the number of repressors. And I found that to be a kind of more amusing way to go after it. But I'm glad you raised this question, because for me, the experiment theory comparison is absolutely not, never about fitting data. Okay? That's never what we're up to. So it's sort of ancillary. Now and then we might try and do a data fit in order to get a parameter that we're going to then use later on, like when we look at looping, and we'll say, hey, we already know that parameter, so it's illegal to touch it again. I'm going to show you this tomorrow. I'm going to show you a bunch of parameter-free, theory-first, zero-free parameters. But I'm going to use this. Like I'm going to say, I knew this already, and I told you about it yesterday. OK, so if you want to talk about it further, we can. OK, so I just want to say, it'd be very interesting to come at this problem from a completely different technique. So now let me walk you through going at it using fluorescence. So I want to, I guess, start with the notion of an astronomical analogy. So coming back exactly to the question of distances of celestial objects and Hubble's efforts. So there's a really important question when you're looking at things that are astronomical. You want to know how far they are from the Earth. And so just for the fun of it, put up your right hand or left, doesn't matter, with your index finger up, come on, and align it to the wall, and then close one of your eyes, and then go back and forth. Right? What happens to your finger? It moves, right? And so if you know the length of your arm, you know the distance between your eyes, you can figure out, basically, how far away your finger is. OK? So that's called parallax. And that works for nearby objects. That's actually how Romer measured the speed of light. First measurement of the speed of light was looking at the moons of Jupiter and the transit time, depending on where we were in our orbit around the sun. So, but that doesn't work for more distant objects. So a woman astronomer, really she was kind of she was treated as a second-class citizen at Harvard, Henrietta Leavitt, in around 1910, she made a super important discovery. And her discovery is shown here. There are certain stars which basically get dimmer and then they get brighter again. They're called Cepheid variables. And the thing that she discovered is that there's a simple relationship between the absolute luminosity and the period. In other words, if you know the period, you know the absolute luminosity. That would be like me telling you that I'm always going to carry around a 100-watt light bulb on my head. The moment I've told you that it's a 100-watt light bulb, you know everything you need to know to figure out how far away I am, right? But if I tricked you and I changed it to 60 watts, then you'd be wrong in your estimate of how far away I was, okay? But so this is it's called a standard candle. And so the reason that I'm really hyped up about this, again, with apologies to the NCBS gang, you've already heard me talk about this, but tough luck, you should have left. So, so the point is that I'm a real hater when it comes to A.U units. Okay? In papers, you see A.U, arbitrary units. That's useless for this endeavor of trying to figure out a number of repressors. So somehow, we've got to find a way to turn intensities into number of repressors. And so that's what I want to tell you about now. So this would be a great calculation. Boris gave you a bunch of homeworks. This is a great homework for you to try and do. I wish I had time to pursue it. But this is an idea that Elowitz and Peter Swain and Uri Alon and Nitzan Rosenfeld and John Young had. And it's one of my favorite all-time experiments. And the idea is very simple. You have n light bulbs. Okay? Each repressor has a GFP. And the cell divides, and on average, each daughter cell gets n over 2 repressors. I should note, this is very similar to what Boris just got through telling you. Okay? This is basically an urn problem. You reach into the urn, you grab one. You grab one, you either give it to daughter 1 or daughter number 2. And you just do this again and again. But the thing is, as you can see here, the partitioning is imperfect. And that means that if you plot the difference in intensity versus the intensity of the mother, you'll get curves shaped like this. And you can prove, so here's your homework problem, you can prove that the difference in intensity of the two daughters, squared and square rooted, is proportional to the square root of the intensity of the mother times the unknown calibration factor, alpha. Alpha is the thing that relates intensity to number of molecules. That's the factor that we want to determine. So let me show you this. So I showed you this movie earlier, but now you can see it again. So here's the concept. At t equals 0, you stop production of red. Now the cell divides. A little bit of red goes to one daughter, a little bit of red goes, or roughly half goes to each daughter. The cell divides again. It divides again. And by plotting, so in terms of data analysis, is a nightmare. So Ted mentioned MATLAB routines. Here you have to follow lineages. You have to know every mother cell and its two daughters. And to get a data point, what you need to do is figure out the intensity of the two daughters, the intensity of the mother, take the difference of the two daughters, square it, and that will be a data point. So here's what that data looks like. So this is daughter 1 minus daughter 2, quantity squared. On this is basically intensity of the mother. The red dots are the averages. The red line is a fit, and alpha is the number of arbitrary units per protein. So that is absolutely cool because it means now we can do an experiment and we can basically figure out the copy number. And here, so by the way, for whoever asked me about fitting parameters, here, what you're seeing is the two methods. So on this is basically a graph that merges this methodology and the partitioning methodology. And this one has no free parameters. You just figure out how many repressors there are. You measure the fold change. You plop it down. In other words, there's no fit to do. Why I like this is, to me, this being a biology experiment in living cells, I was pretty happy with the idea that the methodology in which one uses dilution and the methodology in which one uses quantitative immunoblots basically tell exactly the same story. Okay? So, yeah. It probably would. So maybe you should tell everyone what the mother machine is. Or I can. It's up to, no.
The disambiguation that you have to do with the software.
Yeah, so there's a guy at UCSD, I think he's the one who developed it. And what this is you basically make one-dimensional channels, and you put your bacteria in them, and they grow and divide inside of these channels. And basically, what that means is that all the daughters are lined up. And there's an end, and after a while, the ones that divided off early, they go flowing off into solution. So yeah, I mean, it would be fine. We have flirted with it. The Elowitz guys had a bit of trouble with the mother machine, and so I got a little bit worried that it was going to be 6 months of effort. But it's a good point. So anyway, I guess what I would say is that, again, this is very much in the spirit of what Ted told us this morning. The attempt is you have a simple theoretical idea. You want to test it out. There's tunable parameters, which you can tune both as theorist and as experimentalist. You try to do the best job you can to make the measurement. And you try to have reasonable error bars and so on. And for the moment, I've told you about the success. Okay? And I'm not going to tell you about the failures, although the failures are more interesting in many ways. Okay? So we learned lots of interesting things from when this failed. And if anybody wants to talk about it, I'd be happy to. But for the moment, I'm just trying to show you the idea of what would be involved in trying to actually make a careful measurement of a problem like this. So I wanted to show you another example, which I'm not going to talk about in any detail. But Paul Selvin was interested in looking at the motion of molecular motors like myosin. And so the idea is, right here on my knee, you put a light bulb. And then, the thing walks along. And the reason that they were pursuing this is they were curious about this. Has anyone ever noticed that little kids, when they walk upstairs, they actually do it with one leg in front every step. And then when you get older, you somehow learn to do this. So people had been thinking that there were different models for how myosin moved. And one of them was this. And the other one was this. And then there was also like the kung fu dancer one. And so at any rate, it was really neat because the bottom line is that they could tag one of these legs of the motor, and then by looking at the dynamics, you could actually say that it was this kind of motion, and they were able to basically look at the step sizes and so on. So at any rate, what's my point? So my point is that the fluorescence palette has really been an amazing tool for doing all sorts of kinds of precision measurement. I gave you a little tiny bit of a feel, and maybe I also am just trying to advocate for the idea of cleverness. I'm not talking about any cleverness on my part. I'm just saying people are being very clever. So one of the ways they're being clever is beating the diffraction limit. Okay? Another way they're being clever is getting rid of arbitrary units. These are all the sorts of things that people with physical sciences background can do that are super useful. And at least for me, I really like the sort of aesthetic appeal of good science, and I think they're just gorgeous ideas. Okay, so new topic. So far I told you about the fluorescence palette. Now I want to just say a little bit about energy and force and how people go after those quantities. So I've always been kind of amused by this graph that Steve Quake and I came up with. It was primarily his idea, but we worked hard to figure out something which was kind of fun. And it goes like this. The question is if you look at the energy scales as a function of length scale. Let me give you an example. So it takes a certain amount of energy for me to bend this thing. And now if I make this object smaller and smaller and smaller, I will finally reach a scale at which this energy is comparable to KT. And I remind you, KT is 4 piconewton nanometers. And that's kind of the natural unit of physical biology. Next, if you were to take 10% of my charge and remove it so that I became imbalanced, I would be worth more than any bomb you can imagine. In other words, it's a huge amount of electrostatic energy. And it's very amusing that as you take objects and make them smaller and smaller and smaller, and especially if they're in water, you reach a point at which it costs you nothing more than KT to take charges and send them away. And the best way for you to know this is just to remember your chemistry classes where you got tortured with something called pK. So you must have had that, right? I didn't, but my daughter did, so I got to learn it when she did. And so the pK has to do with stealing charges, for example, off of amino acids. And at first cut, you're like, well, why am I allowed to do that? But in a way, you can see, well, you're paying the penalty of electrostatic attraction, but you're getting some entropy from the deal. And so again, KT, when it's comparable to E squared over L, then that gives you a scale which tells you that the charges can be liberated. So at any rate, what's the upshot of all this? The upshot is that at the nanometer scale, thermal energies and deterministic energies are comparable. Okay? And there's many, many examples of that I think are amusing. So people are working hard to measure piconewton type forces acting over nanometer distances, and I want to say a few words about that. So, back in the day, in the 1600s, Robert Hooke, these are actually literally pictures of his experimental apparatus. I'll remind you, Hooke, he's the guy who figured out F equals Kx and all that stuff. So he did it in this context, which is basically in tension. You hang a weight. And then he learned a lot about torsional springs. Here's one right here. And so on and so forth. Da Vinci, he thought already about fluid flows. And I just wanted to say that, this kind of thing has been, over 3 centuries or 4 centuries, has brought us to the point where people now can measure, angstrom-level displacements of individual molecular motors while they're applying 2 piconewtons of force, or something like this. So I wanted to just give a few words about this. just in case you guys haven't thought about it before. So I'm a big fan of springs. Like I try to find every example I could ever dream up of harmonic oscillators. Like the first time I learned about RLC circuits, I almost peed in my pants. I was so excited. It was just so weird that you learn about mass spring systems, and it all seemed kind of abstract. And then you go learn about circuits, and all of a sudden this weird thing of RLC comes up. Actually, it's kind of related to, again, to what Boris was talking about. The diffusion equation is everywhere. There's no escaping it. So at any rate, there's no escaping springs. So here's the idea in a nutshell. You take a laser beam, you basically send it through the objective, and the laser light gets focused. And if you take a 1 micron size optical bead and you move it off of the focus, what you have is a restoring force that pushes it back to the focus. How many of you have actually worked through this using ray tracing? So you have a bead, and so the light coming through the bead is most intense. It's a Gaussian profile, and it's most intense in the center. Now I move the bead off. So has anyone worked through that or seen the idea? It doesn't seem like that many people, so I'll just draw it real fast. So here's the bead. Here's the light. And the light is most intense here, and it's dropping off in both directions. And so the point is that when light here comes into the bead, it gets refracted. And here some light comes in, and it gets refracted in this way. Now what's the idea? Because light carries momentum, when this light gets bent, it actually has to conserve momentum, and so the bead has its own momentum. And since there's more light here than there, you get a net force pushing it back into the focus. All right? So what I was going to show you here is that if you trap a bead like this and you just watch its position over time, you can track it, and you can use the equipartition theorem in the form 1/2 k average x squared is equal to 1/2 kBt. I'm only doing this in one dimension. But you can use this to work out the stiffness of the trap, because the stiffness is going to be kBt over average x squared. So what you do is you measure how far the bead jiggles. You calculate the variance, basically, and then you put that in the denominator, kBt in the numerator, and that gives you the stiffness of the trap. So, so people have gotten this to the point where it's so good, again, probably many of you know this, but we understand pulling on DNA so well that at this point people calibrate and test their optical traps by pulling on DNA molecules and measuring the force extension properties. It's like Hooke all over again, but instead of a piece of metal, it's a piece of DNA being pulled on. So I just wanted to give you two examples of the kinds of things people do with this. So one of them, an experiment that I really love, has to do with taking a bead to which you attach either an axoneme, if you're interested in microtubules, or a particular structure which comes from the crab. Does anyone know what this thing is called again? It's the apparatus that shoots the spike. The crab's, the lemulus sperm. There's a shooter that's actin. At any rate, okay, forget it. It doesn't matter. So, what is it? No, it's not that. It's like that, though. It's a coiled up spring of actin with a hook at the end. That's really funny. Well, anyway, so you can attach a bead to that bundle of filaments, and always there's one last filament that's out farther than all the others. So then what they do is they bring the filament up against a barrier, like this. And then they watch as the filament continues to try to grow. And as it grows, what's going to happen to the bead? It's going to push the bead back. But by looking at the position of the bead, I can figure out how much force is being applied, right? So first I have no force application. I know where the bead is. Now some force gets applied. The bead moves in the trap. I've already calibrated it. I know it's 1/2 kx squared. So F is minus kx. So depending on how far the bead displaced, one can figure out the force. And so here, what I want you to notice is that you can actually examine the dynamics of a microtubule. The force builds up. The force scale is of order 5 piconewtons. You get growth and catastrophes. Here, this is the buildup of force in actin filaments. And then people are very interested in the composite behavior of these filaments. And so my point here was just to show you that Dan Fletcher and Julie Theriault did another really beautiful experiment where you take a cantilever, this is like 20 microns long, and underneath that actin network is polymerizing. As it polymerizes, it does this. It pushes up the cantilever. But just as if I have a diving board at a pool, we could figure out how to measure all of our masses. As far as I can tell, mine would be the largest of anyone in this room. And we could do it by looking at the displacement of the diving board as each of us stands on it. We'd have to calibrate it first. But once we calibrate it, then I just stand on it and you'd say 100 kilograms. You stand on it and say 70 or 50 or whatever. So what they do is they look at the polymerization in fluorescence, and then they also measure the deflection of the cantilever. And from that, they can basically work out the velocity-force relationship, the stall behavior, and so on. So that's another example. So that was just to give you a little bit of a feel for precision measurement of forces. People like Steve Block, Carlos Bustamante, nowadays, as I said before, they're measuring angstrom-level motions and sub-piconewton forces. It's pretty astounding. OK, I want to finish with sequences are the most delicate expression possible of the phenotype of an organism, and that vast amounts of evolutionary information may be hidden away within them. So that promise has been delivered on. We sequence genomes routinely. My buddy Steve Quake recently sequenced 100 of his own sperm cells with a group of only 2 other people, and for very little money. Okay, so that's the era we live in. So what I want to talk about is not genome sequencing, but rather using sequencing as a biophysical tool. So I just wanted to say a few words about how sequencing works, in case you haven't thought about it before. So one of the classic ways of doing this was to use labeled nucleotides in low concentrations. Okay, so here I have a pot. What I'm going to do is I'm going to examine the polymerization of this molecule I want to sequence. And I'm going to mainly have neutral, if you like, normal nucleotides. But sprinkled in, I'll have a few A's, a few T's, a few G's, and a few C's, for example, that are fluorescent, and also that will stop the reaction. In other words, it won't polymerize past these points. And then what you do is you basically watch, you allow the polymerization to take place, and then you run a gel, and you basically look at the color, if you like, or you use 4 lanes or whatever. I mean, that's kind of the old school way, and I don't want to belabor this. But out of this, you're able to actually read off the sequence. Okay? There's more modern ways of doing this, but I thought I'd just spend a minute on the method that I think is a lot more fun, and that is single molecule sequencing. So this is also from Quake. I remember the first day they did these experiments, they did it with 4 nucleotides. So here's the idea. You take a fragment of DNA. Let's say that's 25 base pairs long. You put it on a microscope cover slip like this, pointing up, and you basically, it's like you take my genome and you shred it into a bunch of little 25-mers. And they're all on one surface. They're all just sitting there. And now what you do is you flow in A's that are fluorescently labeled. And you look in the field of view, and you ask, which of the guys glowed? Because if they glowed, they took on an A. Then you wash out the A's, and you put in the T's. And you look again. And then you say, which guys glowed? It's a different color. And so you do this again and again and again and again and again. It takes a fancy microscope. It takes a lot of data analysis, right? Because you basically got to keep track of all these fragments at once. And when all is said and done, you have the sequence of a bunch of 25-mers. And then you have to figure out how to put them back together again. In other words, if they came from longer pieces. So I'll tell you another one of my teaching stories. It breaks my heart. it accomplished nothing. But one time, and collecting all the books. And there's multiple copies of each book. The book I had in mind was Moby Dick. There's many copies of Moby Dick. And then what you do is you shred them into little tiny fragments of only 25 letters. So here I am, fool that I am, to try and make a point. I brought my beautiful copy of Moby Dick in, and I shredded it up in front of them. And nothing happened. They just sat there. So, okay, if you remember Moby Dick, it starts out with, call me Ishmael. All right, you probably don't remember that, but it starts out that way. And so, sequencing would be, one little thing would say, call me, and it would stop. And another one might say, E space Ishmael with something else. And you got to figure out how to put all that stuff together. Okay, that's a very interesting problem, and I'm not going to touch on it any further. So we know how to do sequencing. People do. Now, what can we use this for? So what I want to tell you about is a group of methods that I'm going to refer to as protect, seek. Okay, that's what I think is really an amazing tool for biology. And let me give you an example, which is the nucleosome. I need to fast forward a little bit just to show you what I mean. Okay, so Ted mentioned it this morning. 3 times 10 to the 9th base pairs is the length of the human genome. 1/3 of a nanometer is the distance between base pairs. 1/3 of a nanometer times 3 times 10 to the 9th equals 10 to the 9th nanometers, which equals AKA 1 meter. So that's how much DNA is in each nucleus. On the other hand, a nucleus is of order 5 microns. So the DNA is highly compacted, and one of the ways that it's compacted in eukaryotes is by virtue of histone octamers. That's what these cylinders are meant to represent. 147 base pairs of DNA wrap 1 3/4 times around the histone octamer, and then there's a, say, 25 base pair linker, and then another one, and so on and so forth. So that's what a nucleosome is. And people are asking the question these days, what is the disposition of nucleosomes in genomes? In other words, is it random, or do the nucleosomes adopt particular conformations which maybe or maybe don't depend on sequence? So I want to use that as a way to introduce you to this protect-seek idea. So here's the concept. First, you digest the DNA, and the DNA, it's only the DNA that's accessible. So, I could cut my belt here. I could cut all this off, but I can't cut this part because it's attached to me. Okay, so you cut all the DNA with an enzyme, and you're left with the DNA wrapped around the histone octamer. And then what they do is they basically isolate these nucleosomes, and then they release the DNA and sequence it. So the net result of that is that you get a readout of what bases are associated with nucleosomes. And the point of this figure is just to show you that people do this for transcription factors. They do it for polymerases. They do it for ribosomes, all sorts of things. This is part of the big data revolution, which is really cool, but it's crying out for statistical mechanics, people, okay, in the sense that there are histograms that show where things are bound, but there's no particular quantitative interpretation of what it all means. So I just want to walk you through some examples of uses of this. You might want to copy down the papers and take a look at them. So this is a very nice, interesting paper that asks the question, where does replication begin on eukaryotic genomes? So Ted told you this morning a very peculiar story, which is the prokaryotic story. There's one origin of replication. In our cells, there's not one origin of replication. There are many, and they're also not always the same. They change. And if you have cancer, then they change even more, and so on and so forth. So basically, what they do is they look for the footprint of where particular proteins are. Those are associated with replication origins, and then you get a genome-wide picture. Another thing that's been done has been to look at where transcription apparatus, such as the polymerase, is found. So I'm not going to really talk about that, but you can go take a look at that. Another one has to do with sequencing where ribosomes are found on messenger RNAs. This allows people to learn things such as where our particular proteins, or sorry, where the ribosomes are paused. Now I want to tell you about work that Justin Kenny did with Ted and with Kurt Callan. This is, I would call it the sort-seek method. And this is really one of my favorite ideas in recent years. So I salute you guys. We're doing it at full force at this point. So here's their idea. You have a promoter region, such as that. Did you talk about this already?
No, I was tempted, but if I didn't,
Is it OK? Here I go.
you'll probably get it right.
We'll see. We'll see if I can pull it off. So you have, let's say you have 100 base pairs here of interest. And what you're going to do is mutagenize, say, at the 10% level. When I say mutagenize, I mean you're going to make a new version of that 100 base pairs, but with 10% of the bases changed. And you're going to do that 1,000,000 times, or 100,000 times. And each time, you're going to put that new mutated region onto a plasmid that drives the expression of fluorescent protein. So now you've got 100,000 different cell strains, each of which has a slightly different sequence. And then the idea is to run it through this fancy machine. Most of you are physics types. Have you heard of FACS before? It's an amazing gadget. FACS, fluorescence activated cell sorter. It's basically, it can measure, I don't know, how many, Ted? Like 1,000,000 cells a second or something? Yeah? OK, I didn't know that. So you have laser. The cells are flowing through really fast. And you can measure the fluorescence of each one as it goes by. And if you like it, you can actually select it and put it in a tube. I was in France last year. I went to a winery in Saint-Emilion. It was so bizarre because the modern wine guys basically have a large flow cytometer for grapes. So they have a camera. The grapes come on this assembly line thing. And then they can tell whether they're damaged, and they send them off. It's exactly the same idea. OK, so now you basically have this very interesting table. What's shown here is the sequence and then the bin of fluorescence that it ended up in. The idea being how much fluorescence was produced. And they had the very clever idea of using the mutual information to basically work out the relationship between mu, which is the bin of fluorescence, and the importance of a given base. In other words, you can base pair by base pair by base pair figure out where binding sites are. And so what we're using it for, just to tell you, which I think is really fun, is I kind of have a dream of wanting to tackle that 2,000 genes in E. coli for which there's no regulatory annotation. So we've used it on a couple already and found information footprints for binding sites, and now use mass spec to fish out the transcription factors. And my hope is we can find a way to do this high throughput. So this is the kind of stuff that comes out of their work. Very, very interesting. You basically can get an energy matrix that tells you how each base pair contributes to the binding energy. That's how the graph that he showed us this morning of all the red and blue dots, I think, was figured out. That's my guess. OK, I'm almost done, so Boris can take over. I just wanted to give you two last examples. Again, I'm just trying to show you sequencing as a biophysical tool. This one I really like, but it's very low resolution. But it's a very cool idea. So, and it's quite related to Ted's talk this morning, but now in the eukaryotic setting. You have a eukaryotic genome. It's got multiple chromosomes. Somebody asked the question, well, physical proximity and genome distance, or genome proximity, they're not the same things. People want to know what part of the genome is close to what other part. So here's the idea that they had. Break open the cells, cut the DNA, and then use ligation. So you've also cross-linked the DNA. In other words, you've got the whole genome, and you cross-link it. So you freeze it in its current configuration. Now you snip the DNA, and you ask it to glue onto whatever DNA it finds in its neighborhood. OK? I don't know how to make a gross analogy that has to do with all of you sitting near each other. But if we cut off your arms or something and then glued you together, then we'd know who was sitting next to whom. So it's kind of like that. Then what you do is you release the DNA and you sequence it. And so that allows you to say which chunk of DNA over here is neighboring which chunk of DNA over there. Very, very beautiful thing. These slopes are results from polymer physics. There's lots to say about that that could be amusing. The exponents depend on which polymer model you're thinking of and so on. OK, one last example is the nucleosome one. Here, I just wanted to show you that, for example, in yeast, people have gone through the whole genome and asked the question, where are nucleosomes found? Again, just to remind you, what's a nucleosome? It's one of these particles that involves 147 base pairs wrapped around the histone octamer. What's the experiment? The experiment is sequence the hell out of it to figure out where nucleosomes are present. OK? So that's the concept of the experiment. These different colors correspond to different growth media. But the thing that I want to call your attention to is this. This, to me, is the most interesting thing to come out of these experiments. So what's shown here is basically the so-called nucleosome-free region. Notice that this is highly depleted of nucleosomes. And then this very characteristic periodicity to where nucleosomes are found within genes. And where this subject is at the moment, it's one of these friendship-ending topics. And where it is at the moment is people are trying to understand the rules that dictate how nucleosomes decide where to go. Is it passive? Is it active? Are there enzymes involved? Does sequence matter? Is it something having to do with one-dimensional Tonks gas? It goes on and on and on. Okay? So I'm basically done. I just want to conclude with a thought about theory and biology. This is like part 2 to my rant. So let me do this with a story, which since most of you are physics types, you probably already know, but it's worthy of repetition, I think, once a week. So this is the data from Dulong and Petit. It's around 1830. What they were doing was measuring the specific heat of metals. Again, conceivably, this is boring. If I called my mom to explain this experiment to her, it would be boring. She would say, who cares? Why would you care about measuring how much heat it takes to raise the temperature of a metal by 1 degree? So, but the careful measurement paid off. Because what they found is that whether you were looking at bismuth or lead or gold or platinum or silver or whatever, that when you did this on a per atom or per molecule basis, the answer was always the same, 3 times R, the universal gas constant. That's cool. It's especially interesting in light of classical statistical mechanics because it's like the first thing you learn is how to use equipartition theorem and deduce 3R as the specific heat. So that's all good until Mr. Weber comes along. So Weber, he decided, I'll try to measure this at different temperatures. And this graph shows his data. So what he noticed is that at low temperature, the specific heat vanishes. And it's cool because it's a crisis in the sense that classical physics has no response. The theory curve is by a certain Mr. Einstein. And that's his theory using quantum physics, basically, or using Planck's constant to deduce the specific heat and to show that it vanishes at low temperature. And so Daniel Fisher likes to say the job of theorists in biology is to be wrong. And my point today was really to say that there's a partnership between theory and polarizing predictions. And when I say polarizing prediction, I mean head on the chopping block, dangerous predictions, not 50 free parameter, ooh, sorry, I forgot, I'll change my parameter predictions, but damn it, this is what I think will happen type of predictions. And then precision measurements, precision being defined as good enough to test the polarizing prediction. It may not mean detecting the Higgs, but it may mean good enough to be able to tell that the repression curve is what you think it is. There's a really nice biography of Einstein by Abraham Peiss called Subtle as the Lord, and there's this really good remark, I think. In order to recognize an anomaly, one needs a theory or a rule or at least a prejudice. Darwin took some heat for his theorizing, and he wrote a very angry, it wasn't an email, but it was a very angry letter. And in this letter, he said, all observation must be for or against a view to be of any service. And then just like Ted and I were doing today, he was complaining about the referees. So he said, these damn guys, if I listened to them, I would just go collect rocks and report on how smooth they are. So he was very frustrated because he had an idea and he was trying to think about it in the context of data. So I'm all about being totally prejudicial, a priori, going in. And that's what you heard from Boris a little while ago. It's being prejudicial about what you expect will happen. So precision measurements, I would say, are needed to know if we should even be surprised. So I showed you a graph this morning that had the periodicity of repression as a function of length of DNA loop. And I challenge you, I don't think you knew whether or not to be surprised by where the peak is. I'm just telling you, I'm super surprised. You couldn't be surprised unless you had some sort of a reason to be surprised, which is a theoretical prejudice. Okay, and my hint for you is that that length is very bizarre given the persistence length of DNA. That's the reason I'm surprised. Okay, so what do I want you to remember from all this, besides that you're happy that I'm done? I guess it's now exquisitely possible to measure sizes, numbers, concentrations, forces, timescales, expression levels, blah, 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 with exquisite precision. Theory needs to be commensurate with the experiments. Precision measurements do not lend themselves to loose verbal descriptions. So if somebody's going to go to all the trouble to make a careful measurement, I'm sorry, I'm going to just say it again. I'm not accepting of the idea biologists already know that transcription factors are titrated, or astronomers already know that the Earth goes around the sun. Because let's take a poll. Which is cooler? Which is more important? Copernicus and his insight that the Earth goes around the sun, or Newton figuring out inverse square law, tides, eclipses, everything. Where I'm pretty sure I've already revealed my cards, even though I love Copernicus, okay? Newton. All right, you guys, I want Copernicus? All right, good. What's your thinking, just for fun?
I guess the first step is very hard. First sort of,
I totally agree. I totally agree. Yeah, so in a certain sense, it's kind of a silly exercise.